My name is Kathryn Vanden Hoek. I am an undergraduate student at the University of Missouri-Columbia, working to help improve our understanding of abiotic stress response in plants. Salt stress is an increasing issue for agriculture and is making it even harder for production to keep up with the rising population demands. Additionally, human activity is increasing the amount of toxic heavy metals in the environment, negatively affecting soil fertility and entering the food chain. This problem calls for a safe and effective way to remove contaminants out of the soil. That's where the star of our show comes in. Kathryn is a member of the mustard family that has been shown to tolerate both salt and cadmium. Kathryn's close relation to Arabidopsis and brassica crops leads us to believe that this plant will be a good model system for better understanding phytoremediation and could provide valuable information for the genetic engineering of brassica crops, which will become even more important as soil pollution increases as a result of climate change. So this leads us to two main questions we want to focus on. Primarily, we want to learn more about the mechanisms of salt and cadmium uptake and tolerance in Kathryn and any potential overlap in these mechanisms. Additionally, we're interested in if the Brassica polyploid event plays a role in the increased abiotic stress tolerance of Kathryn. We're going to start by looking at Kathryn's response to salt and cadmium. This is our workflow for the ionomics portion of our project. We grew Kathryn in a hydroponic setup and created 4 stress treatments by adding salt, cadmium, both or neither to the tanks. In collaboration with the Bioinformatics and Plant Sciences program, I worked with a computer science student and we created a real-time tracking system to monitor the conductivity and salinity changes during each treatment. We also sampled plant tissues and ran an elemental analysis to get more information on where the elements were going and when. Here I have shown the data we gathered from the conductivity sensor. While the conductivity of the solution is higher for treatments with salt, we saw a cyclical pattern for every treatment. We tested the sensor in distilled water and got flat readings, but we have yet to test the sensor against hydroponic solution that does not have a plant growing in it. Our working hypothesis is that at different times of day, the plant is excluding ions and taking in more water, and then vice versa, but we can't currently be sure if this pattern is a result of the plant or a fault in the measuring device. If anyone has worked on something similar and has some insight into this pattern, I would love to hear more in the comment section of this video. We also started gathering preliminary elemental analysis data to get a sense of the patterns we will see. So far, we see that when Kathryn is exposed to elevated levels of salt, the concentration of sodium increases in the leaves, increasing in the younger leaves more readily than the older leaves. As far as potassium, we see elevated levels when the plants are exposed to salt, a known trait of salt stress response in plants. We also wanted to look at iron levels because iron uptake genes can be affected by cadmium. Given this current data, we do see a pattern suggesting that stress treatments involving salt cause a temporary increase in iron concentration. However, this is just preliminary data and we are not yet sure of the strength of it. Additionally, we looked at zinc concentrations because cadmium overtakes zinc transporters, but we're not sure if we can take anything significant from the data that we gathered. When we look at cadmium concentrations, however, we do see something we're interested in. Cadmium is accumulating in the roots, but not entering the shoots. This could be an indication that Kathryn uses an exclusion mechanism to deal with the elevated levels of cadmium. We are also noticing that cadmium accumulates in the roots faster in the treatment without salt, so perhaps salt is slowing the uptake of cadmium. While we did see some interesting patterns, it's important to note that this data was taken at only 3 time points, 0, 24, and 48 hours, something we plan on changing when we rerun the experiment. Additionally, there's a chance that we did not collect enough plant tissue to get very reliable data for our elemental analysis. I'll talk more about how we plan on changing the experimental design towards the end of the presentation. We also wanted to dive into the genomic side of things to learn more about the reasons why Kathryn has an increased ability to uptake salt and cadmium. Shrinkiella parvula is an extremophyte related to Arabidopsis thaliana. When comparing the amount of tandem duplicates assigned to different Go terms, we see a significant difference in the amount of tandem duplicates involved in response to abiotic and biotic stress, with the extremophyte having much more. Shrinkiella and Arabidopsis share a family polyploid event, and Kathryn and brassica share an additional polyploid event, which Sean Thomas, the grad student I'm working on this project with, will talk more about during his talk, so make sure to check that out. This information leads us to wonder, does the additional polyploid event play a role in the abiotic stress tolerance of Cochylia? To explore this question, we took the coding sequence from each of the 5 species listed here and built orthogroups to identify orthologous genes in these 5 species. We then identified orthogroups associated with salt or cadmium stress gene ontology using Arabidopsis genes. Then we inferred gene trees and query trees with at least 2 Cochylia sequences to find topologies consistent with the Brachysi ancient polyploidy or tandem duplication in Cochylia. Including orthogroups with a single copy in Cochylia, we found 255 orthogroups corresponding to just salt tolerance, 202 corresponding to just cadmium tolerance, and 61 corresponding to both. Of the 255 salt orthogroups, there were 181 with at least 2 Cochylia sequences. 46 showed patterns of Brachysi whole genome triplication, 70 of tandem duplicates, 32 of both, and 32 with other patterns. This is one of our inferred gene trees, and a good example of what we look for to categorize this gene tree as having a Brachysi whole genome triplication pattern, where we have one potential polyploid event here that gives us 3 copies of each gene, each set of Brassica rapa and Cochylia randoma being more closely related to each other than, say, the first listed Cochylia gene is to the second one. We found 46 trees showing this pattern. Here is an example of one of our gene trees showing tandem duplication of the gene in Cochylia as opposed to a whole genome duplication. We found 70 trees showing this pattern. And then here's an example of a whole genome triplication with tandem duplicates in Cochylia, and we saw 32 trees exhibiting this pattern. Looking at our 202 cadmium orthogroups, there were 149 with at least 2 Cochylia sequences, 54 showing patterns of Brachysi whole genome triplication, 62 of tandem duplicates, 15 of both, and 18 with other patterns. And then here is an example of one of our cadmium orthogroup gene trees showing a Brachysi whole genome triplication pattern. We found 54 trees showing this pattern, 62 showing a tandem duplication pattern, and 15 showing both. So what to take of all of this? We see some overlap in orthogroups for salt and cadmium stress response, and orthogroups with Cochylia duplicates show topologies consistent with whole genome triplication, tandem duplications, or both. Although these topologies are consistent with the Brachysi polyploidy, it's possible they're the result of other duplication mechanisms. Overall, these results suggest that genes resulting from ancient polyploidy and tandem duplications may influence the salt and cadmium stress response in Cochylia. I believe we have a good starting point for both questions 1 and 2, but there's a lot more to do. We want to scale up the experiment, having more samples and testing them for a longer amount of time, as well as adding an earlier time point to capture data on the plant's initial stress response. While so far we have started looking into the ionomics and genomics of the plant, we are in the process of expanding the multi-omics approach and revising our experimental design to look more into transcriptomics, metabolomics, and ionomics to create a more well-rounded picture of Cochylia's abiotic stress response. Once we get a chromosome-scale genome, we can get better evidence to see if these Cochylia genes are actually derived from ancient polyploidy. This work will be important for better understanding salt and heavy metal tolerance, which will be useful for expanding phytoremediation and advancing genetic engineering to help us combat the increasing threat of soil pollution. I'd like to give a huge shout out to PhD candidate Sean Thomas. He has been leading this project and has been a great mentor for me. I'd also like to thank Tasha Ogati, my bioinformatics partner, for helping us track salinity data around the clock, Dr. David Mendoza and his lab for all their help with ICP prep and running elemental analysis, my PI, Dr. Chris Pierce, and everyone else who has and will continue to help us on this project. Thank you for listening, and I hope you have a great rest of Botany 2022.